We're gonna be like teacher. I'm reporting it. Okay. Does that mean?
Okay. We're back. Yeah. Just there's no telling how the rest of the day is going to go, so just go with it, right?
We were gonna let you get away with it. You were staring at
Yeah. No, I would have figured it out. Don't know when I would have figured it out, but I would have figured it out. Okay. So back to work. So temperature. So this one will denature proteins as well. What's our example here? Cook an egg, right? So the egg white, what does it look like when you first crack it out of the shell? Clear, right? So that clear is mostly protein, right? So it's albumin. Remember that one? We use it for our protein source for the macromolecules lab. And that's the one that's in blood plasma. That's your dominant protein, your blood plasma. It's also an amniotic fluid. It's pretty much everywhere in animals. So it's a pretty ubiquitous protein. So if I were to drop that into a hot frying pan, what does that egg white look like after a few minutes? Yeah, can you see through it still? So what happened there was you basically unraveled the proteins. They got tangled up, creating that opaque mass of protein. And so that's what temperature does to it. Okay. Now, salt concentrations can also denature proteins, but you generally, it's a little trickier to do that because we regulate salt very, very heavily. And so it's very difficult to get salt concentrations in concentrations big enough to do damage to our proteins. So basically then all you're really doing whenever you do pH or temperature is all you're doing is just unraveling the protein. So think about this for a second. So all you do is unravel the protein. So if you were to eat, say, a piece of raw, like just a regular piece of protein, like just an unfolded protein, undenatured protein, and you swallow it, where does it go? And what's the pH of your stomach? And what's going to happen when it hits that stomach? Yeah. And then your body goes to work on it, right? So then when you cook the egg in the pan, what are you doing to it? Okay, so wait a minute. And then the proteins will denature, right? They fall apart. So go ahead, Chris.
Does it kind of dehydration versus eating, like in the sense of like warm temperature on the dehydration versus dehydration? If you're cooking the egg, you're right, dehydrating it to an extent.
Are you meaning to use the word dehydration?
Maybe not.
Or are you trying to go for this word? Denaturation.
I'm trying.
Are you talking about unfolding a protein?
Well, when you brought up the point of like,
Because dehydrating is losing water.
yeah.
Is that what you're going for?
Yeah.
Okay. No, dehydration is not a thing. We don't really care. But I do like what you just brought up, though, which is my next point. Is if your protein, your egg, is going to denature before your body gets to work on it, whether you cook it in a pan or you unravel it in your stomach. If it's all the same, then why do you cook food? What? You're probably not alone. I don't think I'm gonna be dancing with a raw egg anytime soon either.
My dad
Yeah, some people do, right? So here's the question. Does it matter? Oh, I hear some no's and I hear some yeses.
Cooking, I don't, the bacteria and stuff that are living in it, which like salmonella specifically for eggs, which is.
But doesn't matter for the actual egg. So eating it raw versus cooking it, because it's all going to denature eventually, right? If you swallow the egg raw, it denatures in your stomach. If you denature it first, it'll be denatured, but it's not that big of a deal to denature it. I mean, it's quick like that.
Well, but the human body more easily digests cooked food than it does raw, no?
No, that's not true. That's not true. You know why? Because we're the only mammal, we're the only species on the planet that cooks its food. So if that were true, then every other mammal would be sick as a dog. But why? I mean, think about it. Why do we cook food? It's protein, right? Your body is already denaturing a protein if you swallow it raw. If you eat that piece of raw meat, you swallow it, your body's denaturing it because you got a pH of 2. So why cook it? Why not just eat it raw and let your body denature it and you can go to work on it?
Why is it then that they tell pregnant? I said it out loud and it's just like.
As opposed to the alternative, right? Yeah.
People that they can't give off food. Because you're
That's-
more susceptible to the potential bacteria in the raw food.
Bingo. So what is the only reason? By the way, taste is a thing. I'm not going to belittle that, right? We have entire networks based on it. But it's not necessary, is it? I mean, when you see a lion out there gorging on a zebra carcass, do they stop and wait and kind of cut it off and then put it on the grill, get those nice grill marks on it, right? I mean, is that what the lion is doing? No, right? Nobody on the face of the planet other than a human being bothers to cook food. So, and it's not true that raw food is harder to digest than cooked food because it's all protein in the end. There's really no difference. All you've done is unravel it. There's no difference between unraveling it in a pan and unraveling it in your stomach. It's all just unraveled eventually. So that's not true. But that's not because it's raw. That's probably has something else to do. I mean, that's probably the entire psychology of feeling something bloody and slippery sliding down your neck. And then you probably have other visions. It's the texture thing with the egg, right? Most of us can't get past the raw egg because it feels like a little loogie, or a big loogie in this case, going down the back of your throat. Slime ball. Yeah, and you can't get past the texture, but that's all psychological. No other mammal on the face of the planet has that. I opened up a can of worms, right? Okay, we've got a question.
So wait, why is it part of the hangover cure?
What?
Why is like raw egg part of like the hangover cure?
There's a lot of mythology in fitness and nutrition. Yeah. Placebo effect's the real deal, though. The placebo effect is real. What you're actually doing for that is not. So, and industries know that. Good. We have a question on the floor.
Like I feed my dog raw meat. Is there like if you let them cook meat?
They digest it just the same. It's all the same. There's really nothing that's happening. It's just a matter of denaturation. It's, do you denature it outside of your body or inside of your body? No. Nope.
They're concerned. So why are the fitness buffs freaking like
the fitness buffs do all sorts of wacky, crazy things that have more to do with superstition than science. Yeah. Well, there's actually a lot. As a matter of fact, I spent a lot of time in AMP having to debunk all the squirrely crap that the entire fitness industry is wrapped around, because those people are absolutely insane. And there's a good chunk of what they do that is absolutely dumb, just straight up dumb. And it doesn't have any kind of physiological sense to it at all. Amazing, right? Darn it, I saw it on the internet. It's got to be true. Yeah.
Are we as humans,
Okay, question on the table.
are we as humans more susceptible to these diseases then? Because like you said, no other species like cooks their meat. So like as human beings, we're just more.
We're different. Right, we different. So just to not to drag it out, but I wanted to beat that one down a little bit, right? To sort of divorce us from this concept that we have to cook food at all. We don't. The only reason why we do is for what? And you made the point a couple of times.
Yes.
There's only one reason, and only one reason, why you need to cook your food. The bad stuff that comes to get us. So the proteins we're trying to denature aren't the food proteins. It's the proteins associated with the bacteria that can make us sick, which is the reason why the mothers are told not to eat anything raw. Not because there's anything wrong with the raw food, there isn't. It's because of the critters that are in it that can cause problems. And a pregnant mother has a fetus that doesn't have the same kind of immune shelter that the mother does, and probably is going to be much more susceptible to harm in the event of like a food poisoning scenario.
I apologize for that question, but like, does denaturation, like, does that have anything to do with actually like the removal of like the bacteria and the bad stuff in the?
So like if you want to go salmonella in the eggs, right? The reason why you cook the eggs and the reason why you get all those warnings, like if you do like eggs over easy, is it's the high temperature when you're cooking the egg denatures not just the egg, but the bacteria, the salmonella that's in there. So you kill the salmonella, you unfold the salmonella's proteins, and it's a dead organism. That's what you want. So you'd still be eating salmonella proteins. It just, it can't do anything to you because they've been unraveled because you cooked your food first. But if you swallow the egg whole, then you're going to be dancing with a live organism that can do you harm. Well, we do. Yeah, we don't. I mean, in some cases we do, but our body has to go through that, right? So if you've never had salmonella before, then your body doesn't know who it is, and none of that. But you don't. Even if you make antibodies, you don't retain a titer in your system necessarily. And generally speaking, you can lose that immunity over time, and it's like you have to start off fresh. So it's like you use a tool once and it's like, I'm never going to use you again, so I'm just going to get rid of you, and if I need you again, I'm going to go rebuy you, right? That's kind of what it's like, which is the reason. And there's certain things that are like that. There's some things that we keep a memory of for the rest of our life, like chickenpox, for instance, right? And then there's some things that we don't, and then there's a whole bunch of things in between.
That's why you have to get updated vaccines.
Yes, that's why you have boosters, right? Is because your body doesn't hang on to the immunity. How many what? I don't know. At least 3.
Also because it's a virus that
yeah.
keeps getting new strains.
It's going to be like the flu shot, basically. It's going to be an annual sort of thing, because that's what the endemic sort of thing is. So, yeah. Any other questions before we move into nucleic acid territory? Nope. Okay. So nucleic acids. So the polymer, so the nucleic acid itself is the polymer of this polymer monomer grouping. right? The monomers are the nucleotides. Now, it's like the amino acids, the nucleotides have kind of modular components to it. So every nucleotide is going to have your sugar. Remember, we talked a little bit about that. We said it was a ribose or deoxyribose. And so if you're deoxyribose, then you're going to be the stock sugar for DNA. If you're ribose, you're going to be the stock sugar for RNA. We're going to take a look at that here in just a second. The other major piece of a nucleotide you're going to have is a phosphate group on one side of the sugar. And then on the other side, you're going to have this nitrogenous base. So all of your nucleotides have these two components. So these are common. The nitrogenous bases are what's variable in the nucleotides themselves. So when you look at the nitrogenous bases, then you have two major families of nitrogenous bases. You have the purines, which are basically two fused rings. And it looks very much kind of like the little double dog dishes that you get from Purina. That's actually how I remember Purines, Purina double dog dish. So it's got two fused rings, the big one and the little one, right? The food bowl and the water bowl fused together. Or if you're like my dog, it's food and food. Who needs water? Right. So those are purines, and we have adenine and guanine are the two bases in that group. And then we have a different structure, the pyrimidines, which are one ring. And this accounts for thymine, cytosine, and uracil. Uracil is RNA only, and thymine is in DNA. So these two swap out when you're in DNA versus RNA itself. Now, just like we learned about the bonds that connected the previous macromolecules, right? So carbohydrates were connected by what kind of a bond? The name of that bond. There's three different types. You had a 1,2, 1,4, and a 1,6. What? Not glycogen, close though. Not hydrogen. No, that's the next one. Hang on to that one. For carbohydrates. Holding the starch together. So the starches are held together by 1,4. You probably remember the slide because I did a slide insert and I wrote all three of those on there. Glycosidic, right? So the bond that holds carbohydrates together is the glycosidic bond. There's a 1,2, right, which holds the hexameric one with the pentameric one, right? So, and then there's a 1,4, that's the one that holding starch together. That's a 6 and a 6 held together. And there's a 1,6, which does the branches on the side. So those are glycosidic bonds. Amino acids are what? Glycosidic. So the amino acids are held together by what kind of a bond? Not glycosidic. They're held together with the name of that one. That was where, yeah, poly means many, peptides, peptide, right? The peptide bond holds two amino acids together. And this one is held together by the phosphodiester bond, which sounds really scary. It's okay, because an ester is an actual another functional group that we study in organic chemistry. But all an ester is basically a carbon, kind of carbon-oxygen carbon bond. That's what it is. That's all it is. And a phosphoester is a situation where a carbon is bound to an oxygen, but it's an oxygen that's in the phosphate. Remember the structure of a phosphate group, where you had these 4 oxygens on there? You're double bound to one of them. So if you do a carbon bond to one of these, that creates a phosphoester. So it's an ester that's part where the oxygen is part of a phosphate group. That's what that means. In this case, it's a diester, which means you make 2 of these, right? This is important because this is how you actually link together your nucleotides. So let's take a look at our nucleotide first. So here you can see your nitrogenous base. And then you have your phosphate group on the other side. Notice it's attached to the 5' carbon. And then at the 2' position, you notice you have 2 different choices here. If you have a hydroxyl group at the 2', then you're going to be the sugar that is associated with RNA, which is ribose. If you have an H there, then you are going to be deoxyribose, and you're going to be a nucleotide that is involved in building DNA. So ribose forms the nucleotide involved in building RNA, and deoxyribose is involved in DNA. So both RNA and DNA have their own bricks. They have their own set of monomers for two different functions. The other thing I want to point out on a nucleotide is what's happening here and here. So at the 3' carbon, you have an OH group. This here is what's referred to as the growing end. That OH is involved in the dehydration. Remember the dehydration reaction? What leaves the dehydration reaction? It's dehydrating. Water, right? Which means that between your monomer and your polymer, your two monomers, you have to have the components of water. So OH is the components of water that leaves during the dehydration reaction. That's the reason why this is your connector. This is the point at which you connect your next nucleotide in line. Melissa, was that a question going up earlier? Okay. So on the 5' side of it, you notice you have your phosphate attached. So this is going to be your 5' phosphate. So this is like the front of your nucleotide, and the 3' OH is like the end of your nucleotide. Think of it like kind of a train. Well, sort of like a train, not really like a train, because you can attach things to both ends of the engine on a train. But imagine this is actually, what's a better one? How about a pickup with a hitch? Where's the hitch placed? On the back, right? So if you want to hook up your 5th wheel to it, it's going to go where? You don't hook it up to the front like a train. You hook it up to the back. So there's a sense of polarity to the truck, isn't there? There's the front of the truck to which you don't connect anything. And then there's the back of the truck to which you connect however much you want to connect, right? The same thing's true for a nucleotide. You've got the front of the nucleotide, the 5' phosphate end, which is the lead, the front of the truck. And then you have your hitch, which is on the back. That's your 3' OH. That's where you add stuff to the back side of the nucleotide. Yeah. The pole, where was that? Like the protein part of it, you mean?
Yeah.
Like when they were bending?
Yeah.
Yeah, so this one's not protein. So it doesn't have the same thing. It can bend, however. It does have some flex in that. So, but it's kind of its own, it's kind of have its own thing because it's a different backbone. So the protein backbone, we saw that, right? We had the repeating backbone, NCC, 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 and then we saw like all the oxygen and stuff like that sticking out on either side of it. We have the same thing here in the nucleic acid backbone. So you have, in this case, you have a phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. And so that forms your backbone. And then sticking out on the 3' end, you have your hydroxyl group. So this is your 3' growing end. And up here, you've got your 5' phosphate. So whenever you have your nitrogenous bases sticking out on either side. So if I drew this like cartoon wise, I could do something like this. So there's my sugar phosphate backbone. And then I have my bases sticking out on either side of it. Now, what are the bases? So here's the purines. So you can see the double dog dish, right? So that's your purina. But one of the things I want to draw your attention to now, again, just like with amino acids, I'm never going to ask you to draw the structure of adenine. That's just not going to happen, right? So, but one thing I do want you to do is I want you to use the knowledge that you already have to sort of take a look and sort of make some inferences about the molecules you see. You know about hydrogen bonds, yes? You know it's needed to be able to create a hydrogen bond, yes? You need some sort of an electronegative atom to create some sort of charge lopsidedness. Usually those electronegative atoms are what? There's two dominant ones that we see in biology. What are the two major electronegative atoms that we see forming hydrogen bonds in biology? OH is O, not the OH part of it, but H is another one, right? Because you've got the negativity, right? The electronegative oxygen forming the negative-ish side of things. The H, which is usually attached to an electronegative atom, becomes the positive-ish thing, right? The other electronegative atom usually that we're looking for is not carbon. There's one that's worse than that. Nitrogen, right? Nitrogen is the other one that we look for. And so we look for these elements, we can see them. Sticking out of our purines, you notice there's this big old nitrogen group, and it's attached to what? Hydrogens, which means those hydrogens are probably going to be potentially positive-ish in nature. We also see an oxygen. We kind of see a little bit of weirdness going on here. So a nitrogen, which probably has a little bit of negativity to it, right? We take a look at guanine. We see a potentially negative carbonyl there. That's a big one sticking out, right? Notice this little NH bond. That's probably going to be positive-ish. This is also going to be probably positive-ish over here. If you take a look at the pyrimidines, then you can see you have a single ring structure, but you have the same thing, don't you? You have this big old amino group out there. These are probably going to be positive-ish looking hydrogens out there. Your oxygen probably going to be kind of negative-ish. Here's another one, NH group. This is probably going to be positive-ish. Same thing you can see with thymine. Probably going to be negative there. Probably going to be a little positive there. You have a carbonyl here, but this is probably going to be negative-ish, isn't it? Because the negatives, carbonyls tend to be very negative-ish in nature, and this is probably positive-ish in nature. Same thing true for uracil. You have a negative there, positive there, negative there, and a positive there. So what I'm just doing is I'm just sort of sketching out where the possibilities are for electrostatic interaction. Now, what do I have now? I've got a group of bases, and they have around them these sort of pockets of positive and negative charge. And what's going to happen when these two get together? They're going to be able to form those hydrogen bonds, right? Now, it doesn't necessarily mean that all of these are going to interact. No, but it just gives us sort of, we're just sketching out what the possibilities are. Right? Later on, we're going to actually write in there what actually happens, right? This is just what's possible. So you can see there's a lot of potential for electrostatic interaction and hydrogen bonding between these bases. Now, the one thing that I want to do is compare DNA to RNA. And so you'll notice, first of all, there's a couple of major differences. Obviously, DNA is a double helix, whereas RNA is a single strand. So you have a double strand versus a single strand. That's one big difference. The other one we've already encountered, right? DNA uses a different sugar. It's deoxyribose versus ribose for the RNA. And then the thymine is used for DNA, whereas that's swapped out for uracil and RNA. As a matter of fact, that's one of the ways you can tell that you're dealing with RNA is if you see a big old U in there, then that is going to be RNA. Because if it was DNA, it would be a T. But which one? Yeah, that's at that 2', right? So for the deoxy at that 2' position, instead of an OH group, which would be ribose, you have an H. So you lost the O. That's why it's called deoxy, because you've lost that O. That makes sense? And that's at that 2' position, what creates that. So the other thing I want to sort of do is I want to finish this out a little bit because we saw some things here, didn't we? Oops, not this far. So notice your double strand, it's not just a single strand, but the double strand is actually zippered together, right, with hydrogen bonds. And what we actually learned is that these bases actually interact with each other and they form hydrogen bonds with each other. So an adenine will form 2 hydrogen bonds and complement with a thymine. It's its partner. And a cytosine will form 3 hydrogen bonds with a guanine. These are what's called complementary base pairs. So for instance, if I wanted to complete this, and I know this is DNA, why do I know this is DNA that I just wrote here? I got a T in there, right? So what if I wanted to complete this molecule? What would complement with A on the other side? Yeah, so I'd form that. How about C? And another C? And A goes with? Nope. That would be a mispair. That'd be potential for mutation. And then these would be tethered to their backbone. And that would be my DNA molecule. So all I have to do then is give it a bit of a twist. And it becomes a little twisted rope ladder. Notice, proteins held together with hydrogen bonds. And shape is critical for proteins. DNA is also held together with hydrogen bonds. Does that make you a little nervous? Good, Ricardo. Oh, there's a lot of them. Actually, in the genetics class, which I'm sure you're all going to take in the fall, I'm going to start my pitching now. But we have an entire chapter just on mutations. On one of the types of mutations that you can get is what's called mismatch mutations. Those can come from multiple different places. Your replication enzyme can just make typos. And the problem is when it makes a typo, it doesn't know which one is correct. So if I basically mismatch something, I don't know if that should have been a C or a G. And if I choose incorrectly, then it becomes a mutation. But there are other like chemicals and things like that, which can induce damage and change these bases from one to another. And that would also be a mismatch. But there's like a lot of it. And there's a whole gamut of chemicals that can do that job. But yeah, that's typically, in short, mutations would do that. And there's a lot of different ways to get to that. So nucleic acids, basically, the important thing about nucleic acids, especially with DNA, is these guys are informational molecules. So DNA itself is the master blueprint. In general, nucleic acids are informational molecules. That's what they do for a living. So the idea is, right, because we just came off of proteins, and we know now that the sequence of amino acids is what creates a protein's identity, right? But the question is, how do you know which of the 20 amino acids to pick? So when you're building that long linear chain of amino acids, how do you know which one to pick? You got 20 choices, right? Well, that information is encoded in DNA. So that whole bit of information encodes your proteins. Your proteins don't just come together randomly. DNA actually has the code that tells you which of those 20 amino acids to pick and in what order to pick them. And so that they do that with a sequence of their own. So not only does proteins have a sequence of amino acids, but DNA has a sequence of bases. And that sequence of bases will basically tell you what your amino acid selection should be. Okay, so it's the code, it's the information. We already talked about complementary pairing. But what I want to do is I want to foil DNA against RNA, because remember I said that both of these are informational molecules. So DNA is the master blueprint. So what is RNA then? It's a copy of the blueprint. One of the analogies I like to use for DNA, especially like with the genome, we talked about the genome early on, right? I said there's human genome, which is all 8 billion of us, all our genetic information, all in the pile. Your individual genome is your stuff, your genome, right? Like all of the genetic information necessary to build you from scratch, from zero. And you get that from your parents and from their parents and from their parents and so forth. So my genome is going to be different from Alan's genome, right? Because we have different parents and different families. And so ours is going to be different. So I have in each one of my cells the necessary information to create everything from scratch. Another me, right? The same thing is also true for a master blueprint for any building over here across the street. Or a more complex project that I always like to use, the building of an NFL stadium. Now, it's been a little while since we built Mile High. And that's not a very good one anyway. I want to use my team. I want to use the Raiders for a reason, right? Because Las Vegas it's fairly new. They just built it. And the Raiders did something that no other NFL team has done. I don't know if you watched this process, but they set up like 24/7 feeds on, I think, like Mandalay Bay or something like that. And you could watch the construction site like your entire life. I mean, you could watch the whole thing from the beginning to the end. And then they did something different. They created an entire like documentary video series, basically charting the progress of the construction. And in that video series, they talked to all the architects who were up in all the air-conditioned offices, and they showed everything. They showed their conferences, they showed the programs they were using, how they coordinated all of the work. And there was like so much stuff going on. I mean, there was work, a mind-numbing number of different work groups, all doing different things. And they had them all kind of organized in these really technical programs, and the architects had control of all of it. But one of the key things was when you looked at the 24/7 feed, you didn't see all that stuff at the work site. It started with the diggers, the excavators, who were doing what? They were doing the foundation, right? They were digging a hole in the ground to set the foundation. But it's a stadium, right? So it's not just your average foundation. You're going to have stress points in that are very different. The field obviously probably doesn't need that much of a foundation, but where the risers are, where the people are going to be sitting, you need much more substantial foundation work. So you're not just digging down different layers. And it was kind of cool because you could see a higher, shallower layer of the foundation and a deeper layer of the foundation. But then they had to start digging the individual footings, like pylons, like piers, almost like colonnades, where they would just basically sink these things into the ground. And they had to know how deep to go, how big to make them, how thick to pour the cement, right? So they need to know all that sort of stuff so that they could dig the appropriate foundation setting for the cement guys to come in. By the way, they also had to make accommodations for plumbing. They had to know how to wire in the electricity and everything else that comes along with it, right? So they had to make accommodations. They had to develop elevator shafts. Whenever you see a building, oftentimes the first thing you see going up is this big old elevator shaft, right? So here's the thing. Did that original crew, did they care about what the luxury suites looked like? No, that's not their part, is it? So what information do you think they had at the site? They just had theirs. All they had was a local copy, a working copy of the master blueprint that the architects had up all spread out in a nice flow chart. It was an amazing thing. Vegas knows how to build. I'm telling you, there's no Vegas knows how to build because that's all they do. But anyway, build and take your money. But anyway, right. So they just wanted their local copy for their project, and that's all they wanted. So that was like RNA. RNA is the local copy of the master blueprint for your particular protein. All you need is the information to select your amino acids. That's all you need because you're just making protein X. And so you just need a local copy of that particular information. That's your RNA. The master blueprint, just like the plans for the Vegas Stadium, those stay where? Locked up in the architect's office. They never see the light of day. So when the foremen need information, they run off and a copy of the master blueprint and they say, here's your project. Give it to your people and coordinate them. Right? And the reason why I like stadiums is because there's so much going on in stadiums. There's so many variables in a stadium. It's very, very complex. And that's kind of how you organize it all. But these two work together as informational molecules. One is the master copy. The other one is a local copy that is run off as needed. So that is DNA and RNA. There's one other subset of nucleotides that we can cover next time because this is like the last nucleotide slide, and then we'll dig into lipids.
Speaking of by company as season tickets. So
Nice. Give you an A. But I wanted, if you, and that's recorded, so I can't.